So my name is Klaus Schulten. I'm professor of physics at the University of Illinois, and my group is developing the software VMD and MD that you are going to learn a little bit about during the first 2 days of the hands-on workshop. And so here I am to present the lecture on these programs. So the way these hands-on workshops are designed is that we tell you about the concepts that are realized, scientific concepts that are realized by these programs, a little bit about the algorithms that the programs use, and then we give you some examples describing how the programs are actually be utilized. And these examples are heavily spiced with philosophy, so to speak, because I want to train you in getting the proper attitude about using this software. And this attitude is mainly that I want you to think as you use the software. So the other extreme that I'm absolutely staying away from is that I give you some recipes how to use the software. So this is not a cooking class. we want to constantly be very conscious about the use of the program, limitations, and so on. And so basically, I want to when you ask me questions, I want to guide you about finding the answer yourself, so that you are going through the process of judging yourself what you might be able to do with these programs or not. So here is my presentation of the workshop that I had planned to give a little earlier this morning. And so let me start it. And so historically, we always begin with the group picture of the last workshop. So the last workshop was given actually in Urbana, assume, I think so, and so you see then there are happy people who learned what you are doing now. And this is actually a good way of introducing this workshop by going sort of to the lineage of the workshop where other people took them, because the workshops go back now over 12 years. The first one was in 2003, and so we have a long series of the workshops where we develop the format and improve the format from every workshop to every workshop. We do this, by the way, through evaluations from you. We ask you at the end of each day to evaluate every day, every lecture, every tutorial, so that we are in a better position to constantly improve the approach that we take here. And so you will also be handed out evaluation form today and tomorrow and throughout the whole week. Okay, then we always try to give you a little bit of a feeling for how the workshop takes place. You see here some teaching assistants, some Imata Kosh, my faculty colleague, teaching, and the teaching assistants who teach. Do we have a laser pointer? And then you see here the beautiful place where you are. Probably you will not have too much opportunity to see it. Pittsburgh. It's quite a nice town. It used to be a mill town, and now it is a beautiful, clean town. It has many good restaurants, and so we ourselves will be taking advantage of it. Coming from a small town, we like to see some real food. Okay, so I show you here. So I show you here the other teachers of the workshop. So we have here first the contingent from Illinois, myself and my wife. My wife is a professor of chemistry and professor of physics, but we do basically the same kind of research, namely computational modeling. And so she will be teaching on Wednesday. I teach today and tomorrow. And then the workshop here is really organized and owned by Professor Yvette Baha. She is from the University of Pittsburgh, and she's an eminent computational biologist. And so she is on the faculty in the medical school here, and so she is actually organizing, or she's directing a center of the National Institutes of Health that is, as part of the activities, developing also software and also training people in the software. And then there are 3 of her colleagues, and I'm pretty sure she will introduce them more closely than I can. And so I just like to say that Thursday, Friday program will be done by our friends here from the University of Pittsburgh. So here's a program, and you see in the morning we have the lectures. I mentioned already that the lectures are conceptual and really asking, what kind of things can these programs really describe, and then how do they do that in terms of algorithms, and then how do you do certain things, like the recipe part, where I want to, in the Socratic dialogue, discuss with you. I hope for quite a few questions from your side, how you actually would approach it. You can ask critical questions. Is all molecular modeling any good? You can also then ask, I have this particular protein, I want to study this particular problem. How can I do this, what you just told us, for my particular case? And so that's what you do. And then we go into dialogue, and I hope I can teach you the thinking part of doing molecular modeling. Then in the afternoon is the most important part, which in this case was in the morning already, namely the hands-on tutorials. So you are now experienced already. You've seen the tutorials, so they are self-study tutorials. When you don't quite finish them, you can take them home and continue there. The advantage, though, is doing the tutorial series that you are working through the whole tutorial, even to the end, where you do the scripting, where things become difficult. When you're at home on your own, you're more likely that you cheat. You never get quite to the end, and the end is where the good stuff is. And so, so maybe you try at least some of the tutorials to go really to the bitter end, force yourself, because your neighbor may realize that you are not only made half of it, and you don't want to be ashamed. And so do it, a few times, and so that will be then, good for you. But you can continue them at home, and all the tutorials, we might have actually too many tutorials that you cannot really even start here. You can take them home. You will find that on our website in Illinois, we have, I don't know how many tutorials, I would say 40 to 50 tutorials of the same quality and the same style as the tutorial that you see here. So they're not crummy, they're also really very, very, very well worked out. And those you can download and learn more. So the tutorials are for self-study. Why do we have them then here in the workshop? You know, you come here to the workshop to learn from us. And of course, if you have then questions as you are working through the tutorials, you saw that hopefully this morning already, you can ask questions. You may first ask your neighbor, then you ask the teaching assistants, and you can also ask the faculty. Now, the faculty is good to ask when you actually do something that's probably very dear to your heart, namely to turn what you learn into applying it to your own research. And so we very, very much encourage that you are doing that. And so we are ready to talk to you about your own research and how you can, you know, solve problems that you have with the software that you learn about. And so please do that. So that is sort of, if you of course don't know your research, you have to come to us. And I'm sitting many hours with the participants of this workshop to talk about actually their particular work that they do at home. And so you might first begin with examples that are just from the tutorial. The examples from the tutorial are chosen just that you learn like a monkey to push the right buttons and not really for the science. We also have something that we call case studies that is available on our website that is sort of a little bit the opposite. There we assume you know the software already and now you would like to see more the science side of it. So it's an example where the science is more important than how to push the buttons. And but then of course you can also have your own problem here, and you can start also work on your own problem. There's nothing wrong with it. There you can also, of course, ask questions and we can help you. And so we really hope that at the end of the week, you are coming home, you're going home as a sort of better person who knows also how to apply the stuff that was talked about here in your own circumstances. But please, you have to ask. So this is a key. But usually participants are not shy, and so sure, if I look at you, you all look very intelligent and awake, so you will probably do that. So the first day is on VMD, a little bit NMD. The second day is then lecture-wise on NMD, where I will be going more into the innards of, namely the physics, because that's a common denominator between all kinds of projects that you may have. And so this is also part of what is in the tutorial, and so that you understand what is actually that you do in the tutorial in terms of the science content, that you understand it. And then afternoon you have then the tutorials again. Okay, so on the third day, when then Lucy Schulten is going to speak, we are changing direction a little bit. First, VMD is a program to, it's really an analysis and graphics program to analyze structures and to draw structures, but at the same time, VMD is actually an excellent sequence analysis program too. And it is much better than other sequence analysis programs in some respect, namely that it is both a structure and a sequence analysis program. So whenever you see, for example, a mutation in what you call the phylogenetic tree that tells you how a particular protein type had a grandmother that then divided up through mutations to make different versions of this protein in many species, you might want to know, there's a change there was a mutation going on, or maybe several mutations even. Where were they actually in the protein? And why might have this organism introduced the mutation? And that you see in VMD, because in VMD you see not only there was a mutation, you see the sequence, oh, something changed, one amino acid changed from this species to that species, but you also see where it changed. And so that you may appreciate immediately makes, of course, much more sense. And so in this regard, VMD is a very good program, and it was actually my wife who had put this very powerful sequence analysis engine into VMD. But now she is also doing something that goes beyond what you normally call molecular modeling. With molecular modeling, we are just concerned with the building blocks of living systems, but what we are often interested in are the living systems. The smallest living system is a cell. There's nothing alive that is smaller than a cell, because alive means you have to repair yourself, you have to sustain yourself, you have to divide, and you have to also collaborate with other cells of the same kind or of different kinds, forming colonies. And so that is what the software that she develops, and that software is called Lattice Microbe, that we will be introduced to it. So that is basically a bridge from the molecular level, where the proteins play a role and their reactions, to the cell, where the proteins diffuse around, the substrates diffuse around that are bringing a cell to life. And so that is what she's describing. This is actually also something that is a strength of the Pittsburgh group. So the Illinois program is called, what is it called? My God, I just said it. Lattice Microbe. Lattice Microbe it's called. And the program here in Pittsburgh is called MCell. And so MCell is, you know, the cells are complicated, and I don't want to give their lectures from Wednesday and Thursday and Friday, so rather refer you to these lectures, but they are sort of different aspects. For example, MCell is very good in describing particular aspects in cells, like, for example, a synaptic geometry and the process in the synapse, where the Lattice Microbe is good for simulating entire cells and even colonies of cells. Okay, so this is then already a little bit a view towards what is going to come then Wednesday and Thursday and Friday. I don't want to go into the detail anymore. And here we have a little bit of a additional point about what will be done here by the Pittsburgh people, namely network analysis. I introduce it in my lecture specifically when it makes more sense to you than that I just open this up here. Okay, so I gave you a little bit of an overview, and now just quickly a summary of the principles of this hands-on workshop. So you have to realize this course is a volunteer effort, so nobody is actually paying us for it. We get actually funds for developing material, but you know, like very, very few people are actually being paid. The most people who are contributing the material are actually graduate students and postdocs, because that way the tutorials are cool. If faculty would make it, coolness factor would be too low. And also these are the people who really use the software all the time. And so they write it really from a perspective of a doer. And so in this regard, it is volunteer that these people are not, they're doing that on top of their research. And also the faculty members are not going to be paid for this, except that somebody pays our hotel costs. So we believe in this, in training. We think one shouldn't have software without tutorial and training, and so we are doing this. I personally have taught, I think now about 1,300 of you in groups of 20 to 40, with, I would say, probably now 30 or 40, now probably more, probably maybe already 50 workshops like this. So I'm really bringing myself here to train you, and I believe in it, and I really like to do it, as do my faculty colleagues. And so take advantage of us, but it is really something that we just do because we feel that's a better way of developing software and giving it to people, to train them also in it, than just assume that you are then on your own when we give you the software. So I said already that the hands-on sessions afternoon are the most important thing. The lecture by themselves are not so important. We want you to teach you to do computational biology rather than hearing about it. And so, okay, so you can ask the lecturers and teaching assistants, and also try to help each other. That it can also be sometimes the quickest way of getting going. Model your own system, I mentioned already, and then I also said, please give us feedback for where you think we can improve, or where you think you like something particular. You say, oh, maybe this is so good, do more of it. And then also, it would be nice if maybe even in a year or two from now, you sent us some feedback back. We got actually quite some feedback from students and postdocs who tell us that workshop made a big difference in their scientific life, and that's of course nice to hear, and that's of course also great for our funding, because we need of course funds to develop the software, and our center for macromolecular modeling and bioinformatics that we have in Urbana in Illinois that is doing the software and training them needs of course regular funding. We are up for renewal, for example, next year, and so getting them, you will be probably too young for giving us feedback, but the ones who did it years earlier, where they said, oh, this was really important, the workshop, that is very helpful in this regard too. Okay, good. And so here are the key actors, the teaching assistants. So Juan and Keith, you met already. And then Joe Peterson and Mike Halleck, they are coming tonight and will be then around tomorrow and a few more days when my wife comes. And then Yi, she, and Sihan are Pittsburgh teaching assistant. They will come on Thursday and Friday, maybe already earlier. Okay, good. So now we got the overview. It's usually nice, except that we are running a little bit late in time, that you also get to know each other quickly. So if we can make it real Telegram style, or did you do that earlier already? Yeah. You were introduced. Yeah. Oh, good. Then great. Then I will go around and ask everybody of you, but probably tomorrow afternoon more than now, from where you are, what you are doing, because I like to know. And then please come to me too to ask about your research. Okay, so good. So now let's begin with VMD. So here we have the VMD program. So VMD is looks like a program that can make pretty pictures. That's true, and I'm not apologizing for it, because making pretty pictures is pretty important for the kind of systems we are dealing with in computational biology, because these systems are very complicated. And we have a tendency to only appreciate about our brain, those parts of the brain that are trained in school, where we're getting grades like an A or B. Those are really important, like mathematics and English and so on. But there are parts of our brain that are not schooled and that are really fantastic. For example, the motor brain, you know, maybe in sport it's a little bit trained, but the motor abilities that are coordinated by the brain are incredible. And the most computational, most intensive area-wise, and if you would translate what the brain is doing into computing power, is the visual brain. It's here the visual area. There are 50 visual areas that are looking at different aspects of visual scenes and integrate them. This is a very, very important part of the human brain, and that you don't go to school for it. Maybe some people go to art school, but usually you don't go to school, but you have that power. And there's an incredible intuitive power there, and I think every neurobiologist would agree with me that the visual brain is the most intelligent part of the human brain. And so you basically, VMD appeals to this part. It tries to activate that part in making you understand the structures you are dealing with. So there's nothing wrong with pretty pictures. We are really dealing here with a very intelligent organ that is basically being activated. On the other side, VMD is much more than a program producing eye candy. It is an analysis program. It can analyze the physical properties of structures. It can analyze particularly also the dynamical properties of structures, and so it's really an analysis program. The best way of characterizing VMD is actually the characterization that was given by the eminent, recently deceased biologist Carl Woese, and he called it VMD is a tool to think. And so that's how you should see it actually. So I'm not just mincing words and trying to sound fancy. Please remember this. VMD is a part where you are connecting your brain to the molecular modeling you are doing. Look at it that way, and not like, oh, let's make a nice picture, but it's not part of your thinking. I will appeal a lot to your thinking brain in my lecture today and tomorrow, but this is the first time I do it. So VMD is really a way to think. Now, the data that VMD deals with are many types. On the smallest scale, we go actually even below the atom to the electrons. And so what the VMD can show is it can show atom by atom representation, but it can also show actually atomic orbitals of electrons. So you see their quantum behavior, how the electrons move in a molecule. And VMD is actually by far the fastest quantum chemical graphics program. So there are these fancy quantum chemists who tell you probably every day at your university how much smarter they are than you as a biologist. But they cannot draw their own orbitals faster than we lowly biologists can do. Yeah, so we run circles around them, and they are using it actually a lot. They also use it actually also for teaching, teaching chemistry at MIT, at Berkeley, and other universities. So, okay. So very elementary chemical things. But then we have also larger molecules like RNA, you see here, protein in the membrane, surrounded by lipids, so that are then a little bit bigger structures where you can have various presentations, as you saw already in the tutorial. And then we have a system that are basically subsystem of a cell already, like an entire cellular organelle with hundreds of proteins that form a certain structure, and you can look at things at that level. And then you can also look at the entire cell. This is actually one of the VMD presentation of the data that are generated through lattice microbe, that program that I mentioned already earlier. And so at all these levels, you have atomic resolution, structure resolution, but also volumetric resolution that VMD can give you. I mentioned also that it has sequence information, and so you have different representations. Like you have here an atom by atom representation that's called licorice usually, and then you can present it in terms of cartoons, and this we call new cartoon representation that shows you here in yellow, for example, the beta sheet character of a protein, and in purple the alpha helix, and in this blue the structured loops of a protein, and in white the ends here, the non-structured parts of a protein that are fluctuating around a lot. Okay, so we are actually considering NEMD and VMD to be actually, to give the simplest idea of what these programs actually can do. We consider them a computational microscope. They show you what the macromolecules in the cell do in a microscopic view that is incredibly powerful, because they can resolve all the way to the atom, even to the electron, when you have that information available, and they can particularly also see the dynamics of these molecular systems at work. And so this is the best way actually. I mean, you know, this is always a simplification, and so this is just like one way of looking at it. But if you want to say, in one word, you want to describe to your grandfather what you are doing when you do molecular modeling, you can say, oh, I use these programs that are really a computational microscope that give us really close views of what is going on at the molecular level in living systems. So now this microscope is what is it made of? It is not made of glass and metal tubes like the normal microscope, but it's actually made of knowledge. And basically you are generating the views by knowing from science the behavior of the atoms that make these cellular macromolecules. From chemistry, you know how, given certain chemical constellations that you find, like certain types of bonds that you find in macromolecules, what kind of forces act there between the atoms. And the forces can be described in terms of potential energies that describe then how the atoms pulling or pushing each other, and how they then move. Like actually, the Nobel Prize in chemistry was given mainly for having developed these kind of potential energy descriptions in 2013. And then once you have now the forces between the atoms, then you can go to physics and describe how the forces bring about motion. This is a famous Newton equation that you might have seen in class. And when you have on the right side the force, on the left side you have some mathematical expression that permits you to calculate the motion. Now, in fact, you really don't do it at the level of the Newton equation, but rather you have to bring it to mathematics and algorithm, and this is the algorithmic form of this equation, where you actually have the time discrete, because you cannot have a continuous time. That would mean that you have very, very, very fine points in time, but you have to jump in time to make it computationally feasible to follow the motion. And so you turn this one into then this kind of algorithmic form that you need to put then, of course, into a software program to carry out the necessary calculation efficiently. And then you put those software programs onto powerful computers, and then you can compute everything. So here you see again the VMD software that is sort of providing the analysis and the view. You see here atomic motion. You see here it's for a buckyball. You see here the electronic orbital motion that is connected to this vibration. So that is, they are really linked to each other, these two descriptions. Here you have the whole lipid protein motion together, organizing what you call lipoprotein particles. You see the ribosome, 300 ribosomes in a cell that are synthesizing proteins in the ribosome. So, you know, this is what VMD can do. And it has, at this point, I think now we are closer to 300. We have about 300,000 registered users. So we have a huge number of users. So the software is really a completely industry-grade software. You know, we of course constantly hunt for bugs in the software, and so you are getting the software for free because it's funded by the National Institute of Health. We are very happy to give it away for free, because this way the program is being used all over the world by many, many, many people. So it's free, but that doesn't mean it's bad. It's really pretty good software. The price of the software is actually many million. So I think we estimated once, that was already a few years ago, I would say probably now just the software development cost about, I would estimate quickly from the earlier, more careful analysis we did, $25 million. So very expensive software. But if you say, oh my God, you know, this is probably another waste, I should write to my congressman. If you calculate now how many people are using the software, and if you just would imagine, you know, you have to pay a yearly price, this is, you know, very high level professional software, then I think the price tag per NIH-funded user, so all over the world, from every part of Africa, we have users. So it's very interesting. I learn a lot about small countries when I go to the website and see where downloads come from on VMD and MD. When we just go to the NIH-funded users, which is, I think, I don't know how many percent, but I would say maybe 5 to 10%. They are paying for the software about $100 per year. And for this software, that's pretty good price. Yes, so I mean, you get this kind of software somewhere that is constantly being updated, you wouldn't get it for that price. And so these are just NIH funded, which of course the National Institute of Health is interested in. The remaining people get it all for free, and there are of course many, many more of those. So really, this money does a lot of good. Okay, so here you see again the little bit closer view of the data types that VMD can deal with. You know, here's maybe the most intuitive one, atomic level description of a lipid and of a protein, a channel, and we put a single-stranded DNA through it. It's negatively charged. If there's a voltage difference, then it pulls DNA through. So that is one way. You see not only can VMD show a structure static, but also dynamic. You see there quantum chemical description of a small molecule. with the electronic orbitals around. You see here a protein now like this, but now in a representation that is that we call new cartoon. And you see also at the same time a cloud, and this cloud is actually electron microscopy measurement of the protein. And so you get electron microscopy density measurement data, volumetric, and structural data more in the atomic or cartoon type. Here you see sequence compared at the same time to structure. This is what I mentioned already earlier. We can see sequences, and we can also see where mutations that arise in the sequence come about actually in a protein. This is a protein that loads tRNA with amino acids, and so we see then there are particular sensitive spots in the proteins, making the proteins work better or worse. And so you see exactly where they are in the sequence and where they are in the structure. Here you see the magnificent view of VMD more at the cellular scale. This is a small cellular scale. We have here membrane, and we have here proteins that are quite mysterious, proteins that are membrane channels that have 7 windows here, that like here, where you have to go into the spherical bulb and then through the channel. And for some reason, the proteins have evolved to filter molecules that want to leave the cell. They are first filtered. They have to pass through this small window, and only then can they leave the cell. And here we have now the view that you enjoyed already of VMD when you are working with it. This is your standard view. You have the main menu. You saw that already. You have an output output window where you can sometimes see amino acid types and other information. Then this one is the most often used representation menu that controls the color of your model, the type of representation you want to use. And here you have a command a module that where you can type in commands to make the script what VMD is doing, to make it do many times the same, or to make it do very complicated things. It answers in Boolean logic about selecting atoms here and so on. So this is what you saw already, not so nicely arranged. These are usually completely independent, and they can be on top of each other. We just arrange them here that you see the main components. And so here you see also what I mentioned already, that we have sequence, a simple sequence viewer, and that just here we have a protein, and here we have the sequence that makes the protein. And now we have, for example, highlighted amino acid 48, and that amino acid 48 is this yellow piece here. And when we would glide with this thing from 48 to 47 to 46, this yellow thing would move through the protein wherever we do it. You can also click on 1 or 2 or 3 and see several side groups at the same time. So this is a very simple, straightforward connection between sequence down here and structure, but you also have the multisec, the tool that my wife developed in her group for VMD, that does much more complicated things. In particular, it can do phylogenetic trees. So if you have many sequences of a protein, let's say you study a protein, and if you're a physicist, you think you're so intelligent that you don't need any more information than yourself. Of course, a big mistake. And if you are more normal, I'm by the way a physicist, so when I beat on physicists, and I'm allowed to do it because I'm myself a physicist. So, but the physicists are a little bit too arrogant for their own good when they go to other fields, and that's why they're a good model to talk about, or a bad model to talk about. But anyway, so when you are a physicist, you just ask your brain, oh, what is going on here? And if you are maybe a chemist, particular biologist, you're more modest, and you say, why don't I learn from nature? And then what you do is you are saying, oh, here's my protein that my professor told me to understand. Why don't I go to the databases that tell me all the sequences that are known for this type of protein? Let's say 100, maybe 1,000, 10,000 sequences. And now I want to know what is in this protein when it works, what is different between the sequences? Where are the sequences all exactly the same? Where is the protein conserved, and where are fluctuations possible? And where are major changes in fluctuations when I go, for example, look at the protein in a bacterium, and in a plant, and in an animal, and maybe other archaea, some other cell types. Why don't I look at that? So you take those sequences, and then you do something what you call a phylogenetic tree. You just like you know, my sister-in-law just got recently retired, and now she got very interested in family history, and so now she studies where all the Schultens came from over the last few hundred years, and so that is what you do with the sequences. So you have these sequences, they arose during the evolution of biology at different times, and now you put them all together. Who were brothers and sisters, and where were the parents and the grandparents? You call that a phylogenetic tree, literally. That's a word that comes from genealogy when you are doing family history. And now you do this for a protein. And then you see where you got major changes, where the proteins developed into groups, and that you can do actually with multisec in VMD. And then you can see where the proteins differ when you go, for example, from the plant and to animals, to archaea, to bacteria, different kinds of bacteria. And then you learn about your protein. You learn where was it conserved and not. So you go into the treasure chest of biological evolution. And there you're getting probably much more information than the most intelligent physicist, I guarantee you. And much faster, and probably also much better. And so that is what basically is possible here. Here you see a phylogenetic tree. See that here you see here here the roots are then where you're getting then bifurcations. These are then you know, parents that get then different children and different children and different children and different children. And this is maybe what you have today here. And here you can present all these proteins on top of each other even. And then you can also show the differences here. For example, these spots here, certain spots here, let's say all the spots in this generation maybe, on this generation, you show the spots where the differences came, and then you realize where the protein changes were made. You can also show where the proteins no changes were made, where it's very conserved and so on. You learn a lot from this kind of phylogenetic tree view. So I mentioned already that VMD can do volumetric data. Here we see again the electrons, how they move when the atoms vibrate. You see that as they vibrate, the electrons move. Here you see actually a very interesting presentation. This is a ribosome, and it has been studied through electron microscopy. Electron microscopy gives the electron density of the ribosome, and here you see from prior knowledge, we knew the ribosome from crystallography. From electron microscopy, we know the ribosome in the functional state, and we see here that an arm of the ribosome in crystallography was used because the ribosomes in the crystal were holding hands. But in the functional form, the hand has to be close to the body because that will work. Here you see the tRNAs that are reading. This tRNA is reading the genetic information from the messenger RNA it reads, and then it finds, oh, this is exactly my messenger RNA, my codon, and I bring you now the next amino acid that is being elongated in this thing. So you just saw there a moment where a tRNA was reading the messenger RNA that is here, and this is all pieced together by taking pieces of from crystallography under artificial conditions and puts them together in a density given by electron microscopy that gives you a functional state picture of the ribosome, and so you can put it together. That's all what VMD can handle, namely puts it into the right shape, and VMD shows it all. So, so then VMD can calculate physical properties. For example, it can assign hydrogen bonds. Where would you expect hydrogen bonds to be in a protein, for example, or in a DNA? It can show salt bridges where you have positive and negative ions coming together. It can calculate distances between atoms. While everything is moving, by the way, it can calculate surface exposed surfaces exposed to the solvent accessible surface. I don't know what the last A stands for. So this is basically where water can hit on proteins, and it can calculate electrostatics, and so it can calculate these kind of properties. Can do the root mean square deviation, how one protein differs from another one, root mean square fluctuation, how proteins through thermal motion move around, and it can present this all to you. For example, show you then when you calculate this property, root mean square fluctuation tells you where the protein moving more or less, and you see where the protein is more flexible and less flexible, and so on. So it really does this kind of analysis very well. Here you see a particularly important thing for modern computation biology. Here you see a protein folding, and you see the protein folds here. You see the shadow of the crystallographic form, and MD is calculating the protein. And you now see we got it not quite exactly here, it's a little mistake, otherwise perfectly folded. And now this is a complicated process, and you want to follow this process. And this process takes actually between microseconds and milliseconds. You have huge amount of data. In the old days, the simulations that we know when my students worked on these kind of things like 10 years ago, their simulations were like typical 10 nanoseconds. So now they are 10 microseconds, 10 milliseconds. They are 1,000, a million times longer. So when those students 10 years ago thought, look at your trajectory, yeah, professor, I do it. Why didn't you do it? Oh, it took me 2 hours to do it. It took a little bit, professor, sorry. That was 10 years ago, 10 nanoseconds. Now multiply 2 hours by 1,000 million. I cannot expect this from graduate. Maybe Keith. Keith is one of those real endurance students. Brilliant and high endurance, but even he couldn't do it. So you need help, and this is what this part of VMD is doing. It can show you the trajectory as a function of time, and it can show you all the amino acids in the protein, and then it can assign here secondary structure. If you have alpha helix, in this case we only have an alpha helical protein, so we only have secondary structure that is shown here in purple. Purple is where we see alpha helical content. If we had beta sheets, we would also have yellow there. Beta sheets are usually shown in yellow, but here we have only alpha helical. Now you can, as the protein folds, you can follow it, and you can then see where things change, and then you can go to every step here and see then the corresponding protein at that moment. Here shown. Here you see the dotted line, and this dotted line corresponds exactly to this protein. So this is an amazing tool. You cannot live without it. And so let me tell you a little bit more about it. And first I show you now a little VMD session. I prepared it because it would take too long to go through the VMD session and loaded it. But so I just like you, this morning I loaded my own VMD session. And so I have to find it though now. Here I think it should be this one. Okay, so here I have the VMD session. Looks actually pretty good. Let me, okay, that's good enough. And so now this is a folding protein. And what I do is I show you the trajectory of the protein, how it folds. And so here we have the protein, and I show you in licorice representation where we see the bonds as licorice, delicious licorice, and the hydrogens here as white, atom as white, the carbon as light blue, nitrogen dark blue, oxygen red. And so now you see how the protein moves when it folds in the VMD program. So you see how it folds. Looks pretty impressive, but you don't see that much really, because it's too much information, too complicated. So then what you do is we stop it here. We can go all the way until it is folded at the end, but you couldn't tell much. So this is why you don't use this kind of detailed information. When you need it, you go to the detail, but what you rather do usually is you're taking much simpler presentations. For example, new cartoon. So a new cartoon is this presentation here, and now we are showing now the folding of the protein in this form, and you will see that it is much easier to follow the folding. See that it goes. Now I want to go a little faster, so good. So go here, and so see it goes on. We go a little faster here, and here we are at the end. We have our protein folded. And now, however, you might want to see other properties too at the same time. For example, you could add this property. Now we see every atom that makes actually this kind of alpha helices that we see there. But that's, again, too messy, so maybe we take it better out. But you see that we can fit it in there. So we take it out, but we can show the surface of the protein. Here's the surface of the protein, and now we see the protein folding in terms of the surface. So now we see how the protein is folding when you just look at the surface. Now then you may say, oh, that's very interesting, but maybe I want to see both the alpha helical form that I cannot recognize here and the surface. The surface here, by the way, is very interesting. The white part of the surface are the show amino acids that are hydrophobic, that really don't like to be exposed to water at the end. The blue ones are positively charged amino acids, the red ones negatively charged, and the green ones are polar amino acids that like actually water but are not charged. The charged ones also like water. So now what you can do is you can change the surface to transparent. Now you can still see the surface, but you also see what's inside. And then you can add actually the hydrogen bonds that stabilize the alpha helices. You may know that much from your biophysics, biochemistry book. Now we start from the beginning again. And so now we see the hydrogen bonds. At the moment, they are usually with water molecules, not within the protein. But after a while, the hydrogen bonds are within the protein, stabilizing the alpha helices. And so you see we have here three attributes: the hydrogen bonds, the new cartoon, presentation of secondary structure, and the surface, all together. And so that gives us then this combined view that is really very valuable, and very often students, postdoc, faculty member work on it for a long time to get a nice presentation, and then they come, say, oh, here, look, professor, look, professor. One can really see something that we wanted to understand very, very nicely in this particular presentation. So it's really worth it. And you see that when you are working on a presentation, you can also see it in the dynamics. So once you do it for one window, then you can say, oh, but show me the same kind of presentation with the same kind of hydrogen bonds, the same kind of distances and so on, all the time doing a trajectory. Here it is. So this is very, very useful and important. Okay, so this is, like here you see I rotated around, so this is not a movie, it's really an ongoing VND session. Okay, so now we stop this session. So, oops, I stop here the trajectory, good, so, and I hide it again. And now I go back to my presentation. And now I want to show you about this timeline program. We call them actually, in VMD, we call the routines that run their plugins. And here we see now the timeline plugin that can follow the dynamics of a protein and tell us about what we see. Here we have actually done the following. We don't fold the protein, we unfold the protein. We are pulling at the ends of the protein one there and one there, and we stretch it out. And as we stretch it out, we initially have a really nice secondary structure, beta sheet, alpha helix, beta sheet, and so on. Actually, here we have only loops, so these are loops, they're not alpha helix, that would be purple. So we have loops and beta sheet, complete. Everything has this kind of secondary structure, but then when we pull it apart, going in time, no secondary structure is left. And now we want to see how, as we pull it, we are losing this secondary structure. That's what this timeline is doing for you. And so it can look for many, many, many frames that would take a graduate student several lives to look at. Now you can put it all in this program, and you can compress it. And if you see something interesting, like here, oh, here we lose this secondary structure, here we lose this one. How does it happening? You go there and you see the detail. And that I want to show you a little bit by now clicking through this thing. We see, and here we have a red amino acid, and this is this red amino acid. The dotted line here is time t equals 0, protein fully formed, and we are just showing one particular amino acids. Here, okay, good. So now we want to, work through this a little bit. What we do is we make the protein a little bit smaller, because when we stretch it out, the protein becomes long, and so it's better that we make it smaller. Okay, so now we see we are going there and we're pulling it. As the protein is being stretched out, as I told you, you see how the protein is stretched and loses all its secondary structure. We keep the colors there, that we remember where were the beta strands and where were the loops, but the structure is really lost. There's no beta strand formation anymore. Okay, so you see how you can very easily go through. Now we have some more examples of the same in a way. So now we are seeing the protein a little closer as we had it before. And now we are showing different manipulations. You see we can fully rotate the protein there in the window, just a total normal VMD window. And now we can sort of go through this timeline data and look at certain points. So now we go to some point in high resolution of the data, and now we click there, and in this moment we see here a piece of the protein that lost its beta strand structure there at this point. And at this point, and see we go here, and now we go forth and back. We see how the beta strand structure is reformed and unformed. And then we can also move up and down the glider to see other amino acids, where they are exactly as this is happening. So I could go, you know, a lot. I think there are many more. I don't want to go through the entire thing, but I think you are getting an idea of what you can do. You know, we just do a little bit more of the same, and you know, I don't want to go through it. What I do is I rather stop here and show you more of these plugins. So here's the list of the plugins. And I will show you the list of the plugins later, but these are the main plugins, actually more plugins. And the name sort of tells you often what they are doing. Sometimes they're a little bit abbreviated, so you must learn sort of what these acronyms mean. But here is the list of them. And then I will show you later, many of them have been contributed by other groups. Oh, sorry, I have to. They've been contributed by other groups, and so they're not only coming from us, but they come basically from people like you. And maybe one of you, when you need a certain feature of VMD, you can program VMD. That's what you're learning when you are going to the end of the tutorial. You can script VMD, and then you can do interesting things with the plugin. Scripts are usually very powerful statements that can do a lot, but they usually do it very slow because they have not written in a real programming language like C++, which is the language VMD is written in. They have a very good script. You make it first available as a script, but then it is, if you have a very good plugin, you make it available as a script. The people use it and they compliment you, but then they will say, oh, but can you make it faster? If you want to make it faster, then you must really go and put this plugin into the actual code of VMD, and there you usually, you can do it yourself. The code of VMD is published every night. Why every night? Because we are changing VMD, and every night, if we change something or not, we are giving the latest code of VMD so that everybody can see what is actually in the program. The same for NMD. So like when we started to do some great advanced work where we simulated much larger systems than everybody could, people accused us of using secret code and said, no, nobody can reproduce it. These people use secret code. And that was total rubbish. They went actually after us at Nature where we published it and at NIH where we get funded. And we could tell them all the time, what a nonsense. Our code is published every day, every night. And so everybody can have it there. And we explain people how to run the program. So in this particular case, for doing very large simulation, we put some extra tutorials on the website so that you know, what people learned how to do it more easily. So everything is available, and this is very important because if you use VMD and MD, you can be sure that what you do can be reproduced. If you're using some other program, there might be the problem that they are not, you don't know yourself what the program is doing because the code is not available. Other people don't know it, and there might be develop a problem. But in MD and VMD, the code is completely open. We have the tutorials, like you experience them now, so that we have an extreme degree of reproducibility. Everybody can use the same program for free and knows exactly what's in the program. Okay, so then when you have a plugin that is really very popular but little slow, then people beg you, and then what we do is we work with you and put then actually your plugin into the core code of VMD, and then it can become really fast because then it's like we are doing also, we know better how to do things fast than even other experienced users know, and so then some codes can become very fast. Particularly, VMD can use graphic processing units, graphic processing units, of course, if the graphic program should use graphic processing units, but graphic processing units can be used today for graphics and for general computing. And VMD uses the graphics processing units, of course, in here in my laptop for graphics, but it also uses it for calculating these properties and calculating them extremely fast. So here you see some examples. When we calculate, for example, molecular orbitals, atomic orbitals or molecular orbitals, I told you we are very fast. We are 120 times faster doing it with the GPUs than doing it without the GPUs. Without the GPUs, we are already faster than the quantum chemistry programs. With the GPUs, we can do it 120 times faster. So really, really fast. Or here we can study how gases migrate through protein 20 to 30 times faster. Electrostatics, very, very important to calculate electrostatics. We can do it for very large systems. And I just say when we use GPUs, we have already a fast solution. Without GPUs, with GPUs, 20 to 40 times faster. So VMD is an ultra fast program. We very, very proud in what VMD can do. Particularly when you go to large systems with a high atom count, like the ribosome has 300,000 atoms. Here the ribosome, that would have been impossible to do on a laptop like mine, but I can look with my laptop and look at the ribosome in no time, very, very fast. And now we have actually another revolution that we will be showing you during the coffee break, which you have to check out in 15 minutes, during the coffee break, where we do something what we call remote visualization. And that is a revolution that was clear that it was great, because when we developed it, it was immediately gobbled up in the group. Very often, we work very hard to make VMD and MD faster, and the group members are last to use it. Sometimes I'm getting conniption fits, saying, "Oh, we did it, we worked so hard, why don't you guys use it?" When we did remote visualization, that was total opposite. They all use it immediately, because now they could do the following. That is what remote visualization is. We have in our group, in our institute there in Illinois, called the Beckman Institute, we have several graphic servers, very powerful computer, much better than my laptop here, and they run VMD really fast and well. So they have lots of GPUs and lots of graphics memory and very fast graphics chips, and everything is the best. Also very, very fast solid state disks, so get data in and out in no time. Now that is where the VMD is running, even though you are doing the manipulation like what I did before at home. The actual calculation for the VMD are all done on the server, and only the graphic stream, the pictures are being piped to your computer, and on the computer you are seeing then the VMD running as if it's calculated on your computer. You don't see the difference, except if there may be a hiccup with your network connection, then you suddenly, the program stops because no new data come. But otherwise, it is not a problem. And we will be showing this. Keith will be showing it to you either before coffee break or after. Just very briefly an example where the program runs actually on the server in Urbana, and here on his laptop, the same as mine, he can do things where you couldn't even load the structure into the laptop because the structure is way too big, and anyway, so basically we are really proud in being big and fast. We are big because we think that cells are big. Cells contain, do you know how many proteins are in your cells? In one of your typical cells, how many proteins you have?
I guess 20,000? Yeah.
20,000. 300 million.
Okay.
If you would be 20,000, you are a very small bacterium. Yeah,
I
so
was thinking E. coli, sorry.
E. coli, yeah, okay. So yeah, E. coli probably have more than 20,000. Don't insult the good E. coli. But anyway, so the cells are full of molecules. If we want to understand living systems, we have to understand big systems. That's where it's a big society of macromolecules, living systems. And so we have to show very large systems because they are very, very natural. Every time we increase the size limits of our program, people said, "Oh, what crazy! You're just a typical male guy who wants to beat your chest and say I can do bigger than you." But no, I'm in love with biology, and I know that biology is a big society of molecules. And if I want to understand the society, I have to be bigger, even bigger than I'm now. Although we reach now a point where size is not really the main issue anymore. There are other issues, but size is not it. Anyway, so we can do very fast things and particularly over very large things. We can of course also do pretty things. We can make very pretty movies and pictures. The pictures are very, very easy to comprehend. Here sort of like a pancake. It's a bioenergetic membrane. The lipids and proteins that contain chlorophyll that convert light energy into the synthesis of ATP. We have here the capsid of an HIV virus. This one you saw already. Here we have a ribosome that synthesizes new proteins into biological membrane. You know, so the picture shows things very, very nicely and clearly. Where when you don't have such nice pictures, you have a hard time comprehending. And so now I want to show you another session of VMD, where it's a technology of VMD that you can use to to communicate with friends in Europe, for example. So here we have a bacterial cell that's looked at with an electron microscope, sliced very, very thin with ion beams. We call it ion beam milling in a slice only 200 angstrom fat, and then looked at it with the electron microscope. And here you see all these rings. When you are slicing basically through, you see rings. But if you go in 3D, you find that all the rings are actually spheres. We just see them different size rings because it depends on where we are cutting through these spheres. And here you see when we are now going and zooming, then we are getting now one of these rings resolved as a sphere. And it's there's this bacterial cell, it's called a purple bacterium, has about 100 of these spheres. And here's a sphere. And the sphere is actually called a photosynthetic chromatophore. It absorbs sunlight and turns it into the energy of ATP. It synthesizes ATP from ADP. And so here you see this one is made of over 100 proteins. And so now we can look at the individual proteins here. We don't show the lipids, just to be a little bit more clear. And so here you see one of these proteins, you see what's inside there. It's an atomic level description. It looks like little cursor, but it's the whole data block has 64 million atoms. And now what you can do with VMDs, what we did is we made a VMD session, and as we made the session, we kept a tape of the VMD motions we did, and we generated what is called a .vmd file. It is actually explained in the tutorial. And so now we went through a session, and we stopped at certain places and made then a point, a stationary point, and we did it many, many times. And now I just show you the stationary point we made, and now you can go send your .vmd file and the structure information to your friend in Europe, and you have it on your own computer, and then you say, oh, click on this button, approach LH2, and then look at what you see. Rotate it a little bit in front of your eye, that you see it a little clearer. You see those yellow ones, you tell them, those are called carotenoids. They are really crucial to repair a cell on the fly when it's being radiated on. And the green things are actually chlorophyll rings that are absorbing sunlight, and they're actually the main containers for the sun energy. And you tell them, you rotate it around, and very, very easy. So now we can go to another one. I don't want to go through everything, but now we go through what's happening here. So now we go to another one. Here we have another particle you see here. Again, we can rotate it. And now we can look at one of them here, this big one. That is actually where light energy is arriving. You absorb sunlight, you turn it into electronic excitation of these molecules, and then it arrives in these bluish molecules, where you are turning then this energy form of sunlight into another form of energy. I don't want to give you the lecture on this here, just to tell you briefly. And you know, let's say at the end you make ATP out of the sunlight, and here you see the ATPase that is doing this, the brownish thing. And so here, this is ATPase, one of the biggest one. Almost every life form has ATP, humans too. Do you know how much ATP you make with this thing every day?
Well, I can make a guess.
What?
It's hard to make a guess.
Yeah, make a guess.
10 billion.
It's you could say, Professor, you are not a very polite man. You shouldn't ask a woman about her weight, because you make your body weight of ATP. So you don't have to tell us in pounds, but your body weight. So very, very important process. And so, okay, so here we see it, and you know we can rotate it in front of us, and you see then every atom there. This is one that's being right now being finally solved in all of its detail, and here you see then how it actually shoots out ATP molecules. So here we see it all together, and so then as the photons are being absorbed, the photon energy becomes electronic excitation energy, and that becomes then different kind of chemical energy, and at the end we make ATP synthesis, but we don't make it. Sorry. Okay, it's supposed to come some ATP molecule out, but it didn't. Okay, good. So let me stop this here, and yeah, I'm almost done. So let me quickly. No, it's a little. I think you're getting a little tired. So I will show a little more. There's very little, but only after we take a breath, and you can ask questions. You want to ask me a question back?
You were showing that long strand protein folding up. So did the software actually follow it? Is it folded, or was it a protein that you had pulled apart and then ran.
Yeah, no. There we just pulled it apart. So it was a normal protein. We stretched it out very correctly with a correct, like you can do it in atomic force microscopy. You're applying a stretching force, you pull it out. We did it about the same time scale, a little faster, but about the same time scale. And with the force that is being applied by atomic force microscope, it's known that it unfolds a protein. And then we just did it in reverse, like showing a movie, you know, like somebody jumping.
So even that first one you showed, that was just a long angle.
Oh, the first one.
Yeah,
the
that
first
first
one.
one.
No, the first one was different. The
Actual
first one
folding.
was an actual folding. So we stretch the protein out to have an unbiased beginning, and then we said protein, go. And then the protein moved around randomly and then became collapsed onto more spherical shape, and then it folded into the right form. So that was a real folding simulation. And that's possible today, and it was quite a triumph of molecular modeling. People were always saying, oh, molecular modeling doesn't really work, and it only works near crystallographic structures. If you go away from it, the force field is no good. And what we learned is that now one can fold proteins quite well. Not in every case, but the mistake you make often, even though it looks like quite a big mistake, is actually energetically speaking very small. So you have then two different forms of the protein, and one of them is more is more a little bit low in energy, and that you call the native form. But if you make a mistake, then the wrong one is a little lower in energy. And so in a way it looks maybe bad, but it's really pretty good, because you have really situations where it have almost the same energy. So, but anyway, so you can fold proteins today pretty well. A protein with, let's say, 100 to maybe 150 amino acids. Okay. So now questions, debates.
Okay. Question. Think of the folding or unfolding from the kind of physics perspective. You just mentioned the energetics, because in the folding movie, I see the extended change is collapsed really fast, like the first couple of frames.
Initially go very fast, yes.
Right, then it went through all that tumbling.
And then it's an entity sometimes find, has to overcome it. It folds almost correct, but then it has the helix in one case was sort of pointed in the wrong direction, the last little helix, and it has to be right, and basically everything goes in 50% of the time, half the time it gets almost correct, and then this one little thing take the rest of the time. That happens sometimes, that there are bottlenecks that take longer to correctly fold, and other things fold much faster.
Right, so I'm curious about to gain maybe some a more either coarse grain or physical understanding, like what type of interactions will work faster, It seems like some type of interaction would be more mistake prone. I would guess it would be hydrogen bonding, maybe they're more prone to mistake.
So what interactions guide this process, and which, if they go wrong, then maybe you're getting caught in a dead end. Yeah, so people have thought about this a lot, but at a time where the computation weren't feasible. So then they developed something like the theory of energy landscape, of protein folding, and they did very, very simple calculations. And on the basis of that, they made many sort of suggestions. Now folding in protein, folding in molecular dynamics calculations at atomic level is only possible since about many years now, not quite 10 years, like 5 years about. And so this is a pretty young field. I don't like to go back to those old days where people used a lot of philosophy and had very poor descriptions. So I think it's better to start from where we are now, where we can now fold the protein, we see the protein is completely stretched out and it finds its crystallographic structure. So it must be right there because it would be an extreme accident that the computer gives actually the structure correctly. And now we want to analyze it. The way you do this is that you realize that the protein is not equally favoring a continuum of many structures, but rather the protein shows very often what you are called clusters. That there's a cluster of structures that it, for example, typically reaches first, and then it reaches another cluster of structures second, and then finally the cluster of structures where the native form is at the center. That would be a very easy case where you have to, let's say, only 3 steps along a linear line. Sometimes it's a little longer, 4 or 5 clusters you go through. Sometimes you also have detours. You have variability. You might go then through different types of clusters. There are different folding routes. And so that is sort of the picture that is being worked out now. But this is again phenomenological. You just do the structures, you do the simulations, you now look at how the proteins cluster, and then you describe it. This is not a very satisfactory situation because, you know, like, as far as I'm concerned, if I show you some amazing reaction, for example, let's say how ATPs synthesize this, how ATPase, the protein that makes our bodies ATP, how that works. And all I can show you the movie. I simulate, here's the movie. But don't ask me any question. Then I would say it's almost useless. The only use is that somebody else might look at it in detail and develop an understanding from it. But if your result of a simulation is only a movie that shows it, and don't ask me anything, that is not contributing to understanding. You just say, okay, we can simulate. Okay, interesting, you know, but that's about it. You cannot do much with it. You cannot remember it. You cannot, you know. So we have to be sure that in simulations we are doing more than just doing it. Like, for example, one of the worst types of simulation are simulations that just agree with experiment. Some people go, very excited. I did the simulation, agree with experiment. What's that? You know, it might agree with experiment in some part and then add new information to it that shows some detail that were not available from experiment. It might also add a new point that experiments have not been made. That it said, for example, if you now simulate this and these amino acids that are far away from the point of action, but they really influence it, they predicted and then the experiment did it, that is what I call real contribution. So just to show a movie is very, very poor. Yes, you have to add understanding to it, and you have to ask yourself very early in your project, what is it that you want to understand? What is what you want to contribute? And agree with the experiment is, you know, you need it to test the simulation, but it is not contributing new knowledge. What is the holy grail of science? The holy grail of science is discovery. You want to discover something. You cannot discover something that's already known. You can only discover something that's not known. If it's experimentally known, good for them, but not for you. Yes, so, okay.
How can we know, after we get the simulation, which range of motions we can really rely on taking results from the simulation, for example? There are fast motions or slow ones. Just looking at the simulation, can you for sure say, we observed this and we observed this very well, we sampled enough, now we can draw conclusions?
Yeah, good. Yeah, so that is a point that I will stress in my lecture, maybe later today or tomorrow. I want to give you a brief answer because you asked a question now, but I give you a longer answer tomorrow. But the point is that the simulation is falling prey to reality. And reality is not easy for many macromolecules. Many macromolecules do not exist in one state. So you know, we say, oh, there's an enzyme that does a catalytic process, and this is a structural enzyme, was published in Nature, and they say they can explain everything about this enzyme. It's usually rubbish, because many proteins exist in several states. In some states, the enzyme is a little bit more active, in some it's less active, in some it is completely inactive. And so when you are, for example, there are some enzymes where they work on a substrate, turn into product, and you can see that process that it turns into product through fluorescence, through optical methods. So they're very, very sensitive. They can look at a single enzyme. Then you see that the enzyme is not doing anything, I'm talking now from that wall to that wall, an hour. So really long time for protein. Now they don't do anything here for a few minutes, and suddenly it's active. And then it goes maybe half activity, then it becomes inactive again, then half activity, then double activity, and then normal quoted activity goes up and down, up and down, up and down. And so now when you simulate, it's the same. The protein, you work very hard, and now you got the protein all together, solvated by water, everything is perfect, and it's not doing anything. And you know, you're getting really upset with yourself. But that's it. The protein has many minima separated by high barriers. So this minima 1, minima 2, minimum 3, minimum 5, and this is one inactive, a little active, very, very active, super active, medium active, inactive. And so you can be caught in one and you don't see anything. But now molecular modeling made dramatic improvements over the last few years by just using our head. And actually the real motivation might have been competition, but a very strange competition. What happened is that we modelers in the science field are funded by the National Science Foundation, by the National Institute of Health, and they give us, you know, grant between $100,000 and $1 million, and $1 million. And then there is, in the United States, a very, very rich hedge fund manager whose hobby is computational biology. His name is David Shaw. And he put several $100 million. He built his own computer, and so on and so on, has an army of programmers in India, and so on and so on. His own power, power plant to drive his computers, and so on. And he can compute, you know, about 100 times, 200 times faster than we can. And now here where we and what could we do? And we remembered our resource as publicly funded scientists and university professors. And our resource is the intelligence of our students and postdocs. And maybe sometimes even a little bit of our own intelligence. And so what happened over the last years is that we developed better sampling methods. So that this guy can just run the computer and it runs all across, and he can run it so fast that once in a while he jumps to the next one, and then he jumps to the next one, but we cannot run so long. And then we so what do? We learned to sample better. And here, for example, we developed a method that was actually a little older already, but that is now being put in all the main programs like NAMD. That is called replica exchange molecular dynamics, where we run several copies of the same protein. One protein is normal room temperature, one is 350 Kelvin, one is 400 Kelvin, one is 450 Kelvin, one is 500 Kelvin. And they talk to each other. And every time they're very similar in their conformation, you can exchange the temperature. What happens is now the hot guy goes zoom over all the barriers. The cool guy goes, hangs here or hangs here, hangs here. But now they can, once in a while, they can exchange, and the result, you are sampling the whole place, and when you are following the rules, then you find out that the cool guys are thermodynamically correctly behaved. They obey, for example, the Boltzmann distribution. And so even though they, by walking up and down the temperature ladder, they are, whenever you catch them at 300 Kelvin, they are distributed in all the degrees of freedom like you expect from a good Boltzmann distributed protein. And so this way we can sample it just like our rich friend in New York. And so we learned other things to use our intelligence to sample better rather than having a computer that is so much faster than ours, so that it can do it just in a totally straightforward way. So the computer that David Shaw, who is, by the way, a very nice fellow, so I don't want to imply anything negative about him. He actually donated even one of his computers here to the Pittsburgh Supercomputing Center, but he only gives us 40 microseconds a year, whereas he can do every year as many milliseconds as he wants. So we can do little steps on the computer, but better that than nothing. But so he's a nice guy, but we can do basically, we learned to go around it. And I will tell you a little bit more about it tomorrow. Okay?
So what is the bottleneck? Is it the accuracy in the force fields, or is it computational power? Or is it both? In finding protein faults.
So the main bottleneck is, you can guess what I said in the beginning. Your main bottleneck is here, thinking.
I mean,
So really, literally. But it's there's in general, I never try to give anybody the impression, it's not you, your brain, it's really like, the computer or the program you use. Use NAMD, it's really a great program. Believe me, it's a really great program. Your brain is more important. There is the Blue Waters computer, the Titan computer from the Department of Energy and NSF funded centers. They are really great computers. Your brain is more important. Don't forget it. So okay, but now where the bottleneck? So there are no simple bottlenecks. Things are not so simple, but the accuracy of the force field is. The force field is doing pretty well. You see that we can fold proteins. Now, when we initially folded proteins, we got sometimes small errors. And the protein didn't fold properly, and that is, we didn't get the energy balance right between beta sheet formation, alpha helix formation. Alpha helix we did well, but when they competed with beta sheet, then we didn't do it quite well enough. And so there was a problem that was then repaired, actually by David Shaw. He realized it from our work earlier that the force field was good enough, and then he tweaked it, very reasonable thing, and then he actually used a modified force field to do simulation where he could then fold more protein than we could, and then we could follow him and could also do it. Presently, there's a big revolution in force field improvement, and that is you call it polarizable force field. So the force field that we have presently they don't permit for atomic polarizability. So if this is an atom with electrons and here comes a charge by, they polarize the one. For example, it's a positive charge and the electrons are being pulled a little bit away and the nucleus being pulled a little bit away, and so you are polarizing this. That makes as far as we can tell, the force field is now working only since very recently fully for proteins. That makes a huge improvement. So polarizable force fields look like they are giving us now much better time, but they will likely not correct everything. The final good force fields will only be quantum chemical calculations that you are doing along with the molecular dynamics simulation. You're calculating all the forces actually in the quantum chemistry program, and that will be likely the ultimate correct way of describing biomaterials. But that is not possible yet. But one can imagine that it becomes a possible route within even only a few years. Because the main point is not so much the computer power, the main point is the human investment in writing a totally new generation of programs. So that was one bottleneck, and computer power. Computer power is also a bottleneck, as I will tell you later, and that is the difference between David Shaw, whom I mentioned, and the rest of us. David Shaw can simulate about 100, 200 times faster, and that way he reaches a time scale of milliseconds. The millisecond time scale is where many biological processes take place. We are good in 10 microseconds. If we are very patient, maybe 100 microseconds, and it takes a long time, and it's still a little too short. So we can only study the very fastest process that happened in macromolecular system, whereas the time scale of a millisecond, many processes take even longer than a millisecond, but in a millisecond you're getting already lots of interesting exemplary processes. So with 1 millisecond you can really keep a whole generation of researchers busy. That is, we don't reach it yet. The other computers are too slow. You wouldn't guess it, because every time you read in the newspapers, oh, there's a very powerful computer was just manufactured by IBM or by Cray, and now used at this Department of Energy computer center in Oak Ridge National Laboratory. Baloney, baloney, baloney. Yes, it's a powerful computer, but all it can do is it can do more calculations. But it cannot do faster calculations. We need faster calculations. More we can do now enough. We can do 100 million, few 100 million atom simulations. We are busy with that. I don't need a bigger computer. What I need is a faster computer. And the manufacturers were constantly selling us bigger and bigger and bigger computers because they didn't know how to do faster ones. And then they were, trying to tell us what we really need are bigger computers. But that was not what we needed. Yeah, so there's a bottleneck in speed to reach a millisecond. That's important, and there's where we have still a bottleneck. But through sampling methods and other tricks like transition path sampling, it's called, or other approaches, we learn to overcome in certain cases this obstacle, and we can basically effectively reach millisecond times. Okay. Good. So, good. Are you ready to show us? So, you know what? I think we deserve now a coffee break, and after the coffee break, you show it? Okay. So now let's. We ran pretty long here. Let's say we have now 20 minutes, and then. Okay, let me continue.